Hello. I guess the title of my talk did a really effective job of scaring people away. As was said, I'm Michael Mayhew. I'm from Duke University, and I want to thank the conference organizers for giving me the opportunity to present our work on a generalized model for multi-marker analysis of cell cycle progression in synchrony experiments. I'm not going to try to convince you that the cell cycle is an important process to study. Without it, we wouldn't be sitting here talking about it. So I'm just going to dive right into the details of how cell cycle works, specifically in the single-celled eukaryote Saccharomyces cerevisiae, or budding yeast. So here I've broken up the process into three phases, G1, S, and G2M. In G1, mother cell starts off with one copy of its DNA, here represented by a blue bar, as well as some other structures that I'll talk about later. The G1S transition, the future daughter cell or bud appears. Over the course of S phase, DNA is replicated and the size of the bud increases. Later on in G2M, in mitosis, the DNA as well as other cell contents are partitioned between the mother and daughter cell. Finally, after mitosis, cytokinesis, and separation of the cell wall joining the mother and daughter cell, the two new cells are free to undergo divisions of their own. So here, the DNA and the bud are examples of what are known as markers of cell cycle progression, and that is features whose status changes over the course of the cell cycle. Now, to describe this a little bit better, I'm going to take our circular timeline, I'm going to linearize it, and we can think then of the bud, in this case, as a marker, and the cells that have them as being in one of two states. Either they have the bud or they don't. That is that they are binary valued. And the window of cell cycle time in which the bud appears here is demarcated by these start and end points, bud plus and bud minus. Now, the important takeaway here is that knowing the status of a marker on a cell tells us something about how far they've gone through the cell cycle. So, how can we use these markers to track the process of cell division? One way to do this is with populations of cells. So how you would do this is you'd start first with an asynchronous culture, represented here by this blue line as being uniformly distributed over the cell cycle. And then by a variety of means, be they chemical, physical, genetic, we can then amass the cells at a particular point in the cell cycle, producing this synchronous culture. We then take the synchronous culture, we release the population into cell division, we take samples of the population at intervals in time, and we quantify our markers of interest. So here at time point 0, under the microscope, perhaps we'd see 0 budded cells and 14 unbudded cells. Later on at time point 30, we see 13 budded and 7 unbudded. And we continue this process until we end up with a series of observations that looks a little bit like this, where each of the data points on this plot here represents the fraction of cells at a particular time point showing a bud. But there are some problems in working with populations of cells, namely that asynchrony arises, either initially because our methods of synchronization aren't entirely perfect, or over the course of the experiment, be due to cell-to-cell variation in rates of progression, or the fact that daughter cells tend to take longer in their G1 phases than mothers. So the goal becomes, how do we estimate the probability that a cell is at a particular point in the cell cycle, given observations of markers? And for this purpose, we previously developed a model called CLOCCS for characterizing loss of cell cycle synchrony. And it effectively does what I just said, accounting for those three sources of asynchrony. It gives us the probability distribution over the cell cycle for the population. And it does this by modeling the process of cell division as a branching process, which I'll illustrate here. So initially, the initial population, represented here by this white normal density, is distributed over a period called GR, in which the population is recovering from the method of synchronization because there might be some stress involved on the cells. As the population enters the cell cycle and subsequently undergoes division, a daughter cell population is introduced. And I'd like to point out here that the peak of the density as well as the width of it, the variance in it, is changing due to these variable rates of progression. And as I just pointed out, there's this daughter cell specific offset. So here we're accounting for these three sources of asynchrony. As time goes on, the population increases, as does asynchrony, such that if we were to look at one of these later time points, our cell cycle position distribution looks something like this, where the probability that a cell is at a point in the cell cycle, somewhere in this range from 0 to 1, is given to us by this mixture of normals. So where do the marker observations come in? Well, what we do is we fit with our model the marker observations by learning first where the start and end points of a marker are. We then evaluate our model fits with a binomial likelihood in which the success probability of seeing a cell with a bud is given to us exactly by the CLOCCS model as the probability that a cell is positioned between the start and end point for that marker. So here we're relating the status of the marker to its position in the cell cycle. So just to sum up, the CLOCCS part, we, via an MCMC parameter inference scheme, learn two sets of parameters from marker observations. The first, on the left, theta, deals with how the population moves through the cell cycle, how long they take to recover, to actually go through the cell cycle, and so on. The second set of parameters, m, deals with when these markers turn on and off, specifically the bud. And here we actually don't learn when the bud turns off, we just assume that to be the end of the cell cycle. But in previous versions of CLOCCS, the only thing we were looking at in terms of binary valued markers was the bud. And if you remember that first slide that I showed you, there are lots of other structures here that we can take advantage of. So in addition to the DNA and the bud, we also have structures like here, the myosin ring, which pops up towards the end of G1 and disappears coinciding with the end of cytokinesis. We also have the spindle pole body, which is a structure effectively like a centriole in mammals that is important for mitosis, that duplicates here at the end of G1S. When we have two of these spindle pole bodies in a mother cell, we call that a short spindle. And when we have one in a mother, one in a daughter, and still an intact myosin ring, we call this a long spindle. So there are additional features here that we can take advantage of. Why would we want to do that? Why would we want to observe more markers? Well, if we just consider the bud, well, if we say we have a budded cell, well, that just tells us that it's not in G1. Doesn't really give us a whole lot of information. But if we start to incorporate these other markers, which have their own windows in which they're visible, we can start to break up the cell cycle timeline more finely and are then able to more precisely say where the cells are in the process. So this is a good thing. So to this end, we went out and performed our own time courses with wild-type strains that had been transformed such that the myosin ring and the spindle pole body were fused to fluorescent proteins and then were viewable with fluorescence microscopy. To calibrate our spindle measurements, we also stained the nucleus with DAPI. So to incorporate these observations into our model to allow us to fit these observations, we introduced for each new marker, for each new binary valued marker, up to an arbitrary number, its own start and end point, which we then would learn from the data. So then our generalized binomial likelihood then becomes something like this, where assuming time points are independent as well as markers, the success probability of an arbitrary marker m sub j at time point t sub I is this p sub j sub I, and again is given to us by the CLOCCS model as the probability that a cell is between the marker endpoints. So I've already described to you a little of our work in making the estimates from CLOCCS a little more precise by incorporating more markers. But there's something else that we can do to do better, and that is we can take into account that certain subpopulations of cells arise over the course of an experiment, and actually, if not properly accounted for, will lead to underestimation of our P sub J sub I. The first of these are dead cells. We put the cells under a lot of strain when we synchronize them, and so dead cells can arise. And what this does to your observations is essentially, instead of looking at a full cohort of living cells with an oscillating marker status, you're instead looking at a mixture in which the first proportion, this 1 minus D, are alive, have an oscillating marker, but the second proportion, D, here have a constant. Here are a constant fraction of which do show the marker over time, but that status does not change. So, considering them in tandem, our observations are bounded from below by the proportion of dead cells that show the marker, and bounded from above by the proportion of dead cells that don't show a marker and never change their marker status. So we learn from our observations not only the proportion of dead cells, but the fraction of dead cells that show the marker. In addition, we account for the fact that the dead cell proportion is not actually growing while the living proportion is. So these bounds, as you can see, are changing with time. The second subpopulation that we want to take account of are what we call early cells. And we call them that because in previous versions of CLOCCS, it was not possible to see cells with a marker at time point 0 or early time points. Essentially, a cell had to get through recovery, enter its first cell cycle, and pass the point at which a marker was visible before this probability could be contributed to the success probability P sub J sub I. But in practice, that's not what we see. So if I show you here, this is an elucidated time course where we're looking at budding. If we look at the early time points, it's fairly obvious that the proportion of cells showing a bud is not 0. And so we'd like to add these cells back in. The difference between them and the dead cells is that they are alive. So they are rejoining the population. Their marker status is changing. And so if we look a little bit deeper into how the synchronization actually works, we can come up with a good explanation for why the early cells tend to be there. So here I start out with my asynchronous population. I expose it to mating pheromone, alpha factor, and the way in which it works is that there's a point in the cell cycle before which the cells will stop cycling and will start to pile up, as I showed you in an earlier diagram. But after which, the cells will continue to cycle. And so, for those cells that are after the point of sensitivity to alpha factor, you need to give them at least one cell cycle to come back around, as it were, and start to amass with their brothers and sisters. Now, this is the more hand-wavy part of biology, in that you have to guess that cell cycle time. You have to guess how much time to leave them in alpha factor. And sometimes you can guess wrong, leaving you with a proportion of cells that might not have come all the way back around, and hence are showing markers. So in order to put these cells back in, we say that there's a proportion of cells from the initial population, so in the point from the beginning of recovery to the first point at which you see the marker, that actually have the marker. What this means is that as the population enters the cell cycle, past that first point at which you see the marker, the proportion that we're calling here that are early gets smaller and smaller and smaller and eventually becomes negligible. It's just in these early time points that it matters more. So here's a few results. Some highlights from what we found. And I should point out that all the graphs you're going to see like this are actually generated from our really full-featured software that is a companion to the paper. So what you're seeing here, the red points are the myosin ring data observations. The purple circles are budding observations. The light green squares are the short spindle. Dark green diamonds are long spindle. And these curves you're seeing are actually 95% posterior confidence intervals bounding our observations. So here for the alpha factor treatment, what you can see is that the observations come on fairly early. Whereas we found that with elutriation, as had been found in previous work, that there was this lag. There was this elutriation-dependent lag because the cells were getting put under a great deal more stress than under alpha factor. So this was comforting to see as it had been documented in the literature. And this was borne out also in our parameter inferences here. So you can see here for the average length of GR that there was roughly 30 minutes more in the recovery time for elutriated cells. Just to show that we could go out and analyze any data set of multiple binary value markers, we got a completely new set of markers from a different lab that actually used a different method of synchronization here on the right column that was actually genetic based. And what you can see in our inferences is the relative differences between these different synchronization procedures. The genetic one, because of its low G1 delay and low GR duration, reflecting more of a synchronous arrest. So CLOCCS also recapitulated the relative ordering of the appearance of certain markers consistent with what was already known in biology. Now, I need to point out that the relative order was known, but that the absolute time at which these events happen is not yet known, or at least is not well worked out. So for both the alpha factor and elutriation, we saw this same pattern of relative ordering. I also should make a point of mentioning how we did in incorporating those different subpopulations, and I'll just highlight the early cells because they seem really to make a difference. On the left, I'm showing the fits and observations for the alpha factor data without incorporating the early cells. And I'd like to specifically draw your attention to the fits here on the right when we do incorporate early cells. You can see that the confidence interval for the myosin ring observations is pulling in these additional observations. The other fits aren't affected as much, but I'll go into why that's important in the next couple slides. At least this is showing us, though, that in taking into account these extra cells, we're adding something back into the model that should have been there. So, as I said, if we look at actually the proportion of cells in this initial population that are showing markers, we can see that the values we get back from the inferences actually correspond well with how the synchronization methods work. So in alpha factor, we see there's a proportion of cells that have a myosin ring, while there are cells that don't really show any of the other markers. In elutriation, however, we see this monotonically decreasing proportion from myosin ring to bud to short mitotic spindle, long mitotic spindle that tracks the relative ordering of the events. And if we look at how the synchronizations work in practice, this is very consistent. So alpha factor, as I mentioned, has a point of sensitivity that is actually right before the point of bud emergence. So you wouldn't expect to see cells with buds, short spindles, or long spindles, but you would expect to see some with myosin rings, which, as I showed in the left column here, is what we see. In elutriation, however, when you're selecting cells that are small, it's not necessarily always the case that small cells means early cells. So you could have some small cells indicated by this tail down here that actually show markers, and they should show markers corresponding to the relative ordering of events. And as I pointed out in the right-hand column, that is what we see. So we feel like this is recapitulating what's actually going on. So where are we going with this? Well, all the observations I showed you are actually done by hand, by someone sitting in front of the microscope. We'd like to get to the point where it's more automatic with smarter algorithms for annotating cells. All the markers we looked at are binary valued. We don't have to use that. It would be very easy to generalize this to markers with multiple values, either converting markers we have or going out and getting new markers. We also looked at the markers independently, which we might want to know which markers tend to co-occur and when. And lastly, on the experimental side, we, for at least some of the markers, want to do a better job, a more precise job of saying what is the particular marker status for a cell. And for that, we want to do better imaging. So with that, I'd like to thank my two PIs, Steve Haas in the Department of Biology, and especially Alex, who's here from the Department of Computer Science, members of the Haas Lab for many painful discussions about the biology, because, yeah, I needed it, and Josh for doing the software, Baoyun for doing the transformations and the microscopy, Diana for work on the modeling, and Shin and Andreas for critical readings of the manuscript. I'd like to thank the ISCB for the travel fellowship that brought me here, as well as these other funding sources. And yeah, I'm going to do the 20th century version. It's a URL. So if you want to download the software, it should be up. Please let me know if it's not. I'd love to get feedback. Thank you.